Good day to you, students. This is the last part of the course that I will be covering, which is amino acid degradation. Please kindly pay attention. There's no note for this, but I've looked at the best explanation I can give for this amino acid. So this topic is very important for you to uh, pay attention to. So I will do my best to explain it. This is a video made by somebody which I think is good. And best practices when you are listening to a video that is like 10 minutes plus, the best is to write everything down out of the forming notes out of the video, so that in case this kind of a question comes out from this video, you will be able to approach it well during the exam. So and I also want to plead with you, don't wait till the dying minutes. You understand, this is the best time to practice the structures, because you'll find structures in them, because in case they may come out. I've also given you an assignment which I want you all to practice. They are questions that you have to submit, and so if you do it, it's going to be a plus for you. So today we are going to talk about amino acid degradation. And why do we need them? Why is it essential for us to look at amino acid degradation? The thing is that amino acids makes up our protein, and so as a result, these amino acids are when they are being degraded, the purpose of this degradation and metabolism of any compounds in the body is to lead to energy production. While there's energy production alongside synthesis purposes, then we also have the major source of energy production in the body is glucose. But in the case whereby there is no glucose is not available for consumption, for conversion. For example, in a diabetic condition or in carnivorous animal, we have surplus of in carnivorous animal we have surplus of protein, because they eat too much of protein. While in diabetic animal, the glucose is not available, so the human body will switch to the metabolism of amino acid. In carnivorous animal, we have less of amino acid degradation, because there is um, because there is sufficient carbohydrate that is able to do what they call it, the energy production. So, and how does your body get the amino acid it degrades? Number one, there are two different ways. The first one is through dietary protein. For instance, when you eat your beans, when you eat your egg, the egg goes through the esophagus, go through the body, the alimentary canal, and once it goes through the alimentary canal, it is being broken down to amino acid. And we all know that the digestion of amino acid begins in the of protein begins in the stomach, and by the time this protein is being acted on by proteases, they form amino acids. Amino acids are the building blocks of protein. So when protein are being broken down, they form amino acid. These amino acids are being absorbed in the blood, into the blood through the intestines, and some these are being transported to the what they call it, which are being transported into the liver, the bloodstream, which are being transported to the liver. So in the liver, that's where your amino acid is being metabolized. We have the hepatocyte, which makes up the liver cells. Now, for your amino acid, you all agree with me that the characteristics of your amino acid is that for every amino acid, there is an alpha carbon, there is an amino group, there is a carboxyl group, and there is a R group. And you agree with me that for every for every amino acid, alpha carbon, amino group, and carboxyl group are always constant. It is the R group that will continue to change, So that's the properties of an amino acid. And so in the breaking down metabolism of amino acid, what we find out is that the amino acid group is being removed, and the remaining which is ammonia, and the remaining component that remain, that is your R group, alpha carbon and carboxyl group, forms your alpha keto acid. So most often time what receive what it forms, what receive your amino group is the alpha ketoglutarate, and this alpha ketoglutarate, once it receives the amino group, it forms glutamate. So you can see in the next reaction that this alpha for alanine transfer is amino group to alpha ketoglutarate, and which is being acted upon by glutamate pyruvate to form pyruvate transaminase to form pyruvate and glutamate. So you can see that your alpha ketoglutarate has been converted to glutamate. Another one is because we have is aspartate plus alpha ketoglutarate. Once it collects the amino group, it forms glutamate and oxaloacetate. This is being acted by glutamate oxaloacetate transaminase. And why is it transaminase? It is transaminase because it is the trans stands for the transfer of something. Trans means transfer. The aminase is the transfer of the amino group, that is the amino group, that's NH3 plus. So that's what is being transferred. And while we have glutamate as the acceptor, we also have amino group donor as the aspartate or the alanine this thing. Now, what determines the product of your, whether as oxaloacetate or what they call it or pyruvate, is because it depends on the out chain of such an amino acid. In this reaction, you will see that another important thing for all these aspartate transaminase is because is the presence of cofactor pyridoxal phosphate, that is derivative of vitamin B6. And you all agree with me, I taught you about cofactor. These cofactors are very essential. So if somebody is lacking in vitamin B6, there is no way the person's body can consume a sufficient amount of I mean, can be able to utilize amino acid for energy production. Now, when we see that our in the hepatocyte, what happens is that we have surplus of glutamate, surplus of glutamate, and this glutamate keeps on forming every time for every amine group, amino group transfer. We have a glutamate that is formed. So for us to be able to maintain the glutamate and alpha ketoglutarate of the body system, there is a back and forth. It is a reversible reaction, and this is converted by glutamate dehydrogenase, which converts glutamate back to ketoglutarate. It converts it converts glutamate back to ketoglutarate, and it requires NAD plus your NAD to form your NADH for the transfer of what they call it, your amine group. And this is what we call deamination, that is removing of your what they call it, sodium ammonia group, ammonia ion from this ammonia ion can be used to form urea, uric acid, which we will see alongside. So this is an example of and trans deamination that takes place in the body. You can see the amino acid which form which trans amino acid and ketoglutarate reacting together to form once the amino acid and the alpha ketoglutarate comes together, amino acid forms your alpha keto acid, and the ketoglutarate is converted to glutamate. So in the body system, to maintain the supply of alpha ketoglutarate and glutamate, this reaction is also how will I put it, see, also has a how would I call it? Is it symbiotic relationship with NAD, and which is converted to NADPH, so that we can easily see that this transfer of amino transfer and the transamination and deamination can take place, so that you will not have a case whereby an amino acid is being degraded and there is no any other alpha ketoglutarate available to be able to do it, because there is a full stockpile of glutamate in the system. So that's why this pathway is very, very essential. So excuse me. So it is also important for you to know that another source of amino acid in the body system, aside the how will I call it, aside the dietary protein, is the body cell. When apoptosis happen, the body releases the body releases its protein content, and this protein content also undergoes the same reaction which I have explained earlier on. So once these amino acids are broken down, the body either do two things to them. The body either use this amino acid to serve as for protein synthesis, formation of another protein, or they undergo further metabolism. So there are two things in the body for metabolism, leading to energy production, and what they call it, protein synthesis, or body essential chemical substance that the body needs. So synthesis occur. So either through your dietary protein or your body cells, the body will make use of amino acids. So in a case whereby somebody, there is a starvation, the body, and there's starvation, that means there's lack of protein, there's lack of glucose coming into the body will start wearing itself out, start consuming its stored protein. It is very important for you to know that for diabetic patients where there is uncontrolled where there is the glucose is not available for metabolism, the body banks on the cellular proteins or protein consumed. So in this state, I've established how the glutamate is formed. So this glutamate that is formed in the body can cannot move within the blood, so it has to be converted. The glutamate has to take in and make up another amino group, that's NH3, and to form glutamine, that's glutamine. Now this glutamine can now travel in the blood and go to the liver. Now when gets to the liver, the liver reverse that reaction by converting the glutamine group to glutamate. And I've established earlier on that glutamate can also be interconverted to alpha ketoglutarate. So this is a deamination reaction, that is removing another amino group from glutamine, so to form what they call it, to form glutamate, and removing that amino group again to form ketoglutarate. So it is very, very possible. And if there is another what they call it, alanine available, the alanine can easily react with alpha ketoglutarate to form pyruvate. So this, let's go back to that first diagram, which tells us that glutamine is being transported down to the liver. So what it does happen is that it's converted to glutamate, and this glutamate is made available for alanine, and pyruvate is formed. Once pyruvate is formed, pyruvate is now used in gluconeogenesis, that is the formation of glucose, and this glucose is made available, and which can be converted to ATP. Also in the muscle, this glucose can be converted to pyruvate and down for formation of ATP. So in case there is surplus of alanine, this alanine is being transported back into the liver, whereby in the liver it's used for gluconeogenesis, or the amine group, because in the process of using the alanine for gluconeogenesis, the amino group, the NH3, is being removed, and when the NH3 is being removed, it is used it is expelled from the body for in form of urea. And that's why we have, what are the fates, the three fates of amino group in the body. We have the amino group. Once it's being removed, it forms ammonia, and fish removes their waste, get rid of their waste in this form. And all aquatic animals, including frogs, toad, shark, and other likes, they release their in forms of ammonia. This is ammoniteric organism. These are organisms. For organism that uses urea, they are called aeroteric urotelic organism, because they release their waste and their amino group in forms of urea. And this is mama, which is also the same thing as bear. For uric acid, this amino group, when it's converted to uric acid, it is most often times that it is being released into the system by uricotelic organism. This urotelic organism are mostly what they call it, they are mostly the birds family. Another name for, what's another name for birds family? Well, that's the end of it all. So what I just, in recap of everything, I want you to understand something, that your for instance, if they ask you for how does the carnivorous animal or the diabetic patient get their energy, you all know that in the state of diabetics, the glucose is not available. The glucose is not available for uh, conversion to energy. So, and in carnivorous animal, they consume surplus of protein, because they eat their flesh eater. So in this case, the body, based on dietary protein, gets its energy from dietary protein, or they get their what they call it, protein from um, um, body cells, or cells, or sperm cells, or that dies through what they call it, apoptosis, or so many other factors that can cause cell death. So now, what how did this amino acid gets to the point of energy production? Now, in the sense that this amino acid, your stomach, once you consume protein, your protein, the digestion of protein starts with this in the stomach, and the gastric juice, the peptases, they breaks down this protein into amino acids. These amino acids are being absorbed during the what they call in the intestine into the bloodstream, and in the bloodstream they are being transported down to the liver. So it is in this liver that the ammonia are grouped. Now, the first thing I want you to remember is that the receptor of this amino group from this what they call it, amino acid, it is alpha ketoglutarate. And this alpha ketoglutarate will react with any form of what they call it, any form of amino acid, to form glutamate, and the uh, resulting uh, resulting compound. So for instance, I gave you two examples. Number one, I gave you amino acid alanine will react with alpha ketoglutarate to give us pyruvate plus oxalo plus glutamate. While in what they call it, for aspartate reacts with um, alpha ketoglutarate to give us oxaloacetate plus alpha keto plus glutamate. This pyruvate oxaloacetate are being transported down to where they can produce energy in the body. Some can be used for gluconeogenesis, for the formation of glucose to be found. In some cases, they find their way to enter the Krebs cycle. I believe you all know the Krebs cycle, where pyruvate is converted to acetyl CoA, and from acetyl CoA we can see it entering the Krebs cycle. And if your pyruvate is even converted to alpha ketoglutarate, it can still enter the Krebs cycle. And these are the ways by which oxaloacetate, these are which by which energy can be produced in the body. So I wish you exam success.